Good evening, everyone. Thank you for joining us for the 2021 Robert E. Campbell Endowed Lecture. Please submit any questions via the Q&A tool at the bottom of your screen, and we look forward to answering them during our Q&A session at the end of the program. Now I have the privilege of welcoming Dr. Stephen K. Labuti, Director of the Rutgers Cancer Institute of New Jersey and Senior Vice President of Oncology Services for RWJBarnabas Health, to offer our welcoming remarks. Dr. Labuti?
Well, thank you so much, Karen, for that introduction, and thank you everyone for joining us today. The Robert E. Campbell Endowed Lectureship was one of the first philanthropic projects I undertook because of the importance of honoring Bob Campbell as Chair Emeritus and his leadership throughout the years at CINJ. Many on this virtual program likely have a connection to Bob in some way. Through his leadership with the Rutgers Cancer Institute Directors Leadership Council, his involvement in the founding of CINJ, as a member of the Rutgers University Hall of Distinguished Alumni, his former role as Vice Chairman of the Board of Directors of Johnson & Johnson, or any other of his many community involvements and achievements. Endowing this lectureship in his honor was a real tribute to Bob's leadership throughout the years. And I want to thank Bob for his support and advocacy for CINJ, and also personally for his support and mentorship to me. And I want to thank all of our friends who made this lectureship possible to honor him. Another project made possible through philanthropy is our Rutgers Big Idea, the Cancer Immunology and Metabolism Center of Excellence. For decades, the cornerstones of cancer treatment were surgery, chemotherapy, and radiation therapy. But in recent years, immunotherapy has emerged as a fourth pillar for the treatment of cancer. Immunotherapy involves harnessing the power of the body's own immune system to fight cancer. There are several immunologic therapies currently used to treat many different types of cancer, some with great success. However, while some cancers respond well to immunotherapy, others don't respond at all. And it's our mission to try to discover why. We believe the intricacies of cancer metabolism may hold the key to answering this question, and by providing treatment for those patients who were previously unresponsive to these types of therapies through a better understanding of some of the limitations of this treatment approach. Discovering and manipulating cancer cell inner workings and exploiting cancer cell weakness, along with powering the body's own immune cells to fight cancer, may help provide the blueprint for the future of cancer therapies and treatments. Thus, the Center of Excellence is at the intersection of cancer immunology and cancer metabolism, and seeks to unite these two disciplines under one umbrella in order to increase research that leads to new therapies for patients, develop cutting-edge facilities to test new interventions, facilitate commercial and university collaborations, and to bring forth a new day in cancer research. Many of the new therapies that will emerge from the Center of Excellence will be cellular therapies, or therapies that are made from a patient's own cells. When looking for a co-director to lead the Center of Excellence with Dr. Eileen White, a world-renowned leader in cancer metabolism, we needed someone who could help us forge ahead in cancer immunology with particular expertise in cellular therapy. That is where our Bob Campbell Lecturer today comes in. Thanks to philanthropic support, we were successful in recruiting Dr. Christian Hinrichs to co-direct Rutgers Cancer Institute's groundbreaking Center of Excellence. In addition to his role as co-director for the Center of Excellence, Dr. Hinrichs joined Rutgers Cancer Institute as the chief of the section of cancer immunotherapy, following his tenure at the National Cancer Institute, where he most recently served as a tenured senior investigator in the genitourinary malignancies branch. His research focuses on tumor response and resistance, with notable accomplishments in the development of cellular therapies for common solid tumors. He also discovered and is developing new treatments for human papillomavirus, or HPV-associated cancers. His work includes the discovery of genes for T-cell receptors that target HPV-related malignancies. Dr. Hinrichs was selected in 2015 by the National Institutes of Health as a Lasker Clinical Research Scholar as part of a joint initiative with the Albert and Mary Lasker Foundation to support talented early-stage researchers in independent clinical and translational investigation. A key feature of this program, which is highly competitive, is the transition of funded scholars who are conducting intramural research under the NIH umbrella to continue with that work at external institutions or extramural institutions. And Christian is the first Lasker scholar to make that transition. With that, I am pleased and privileged to introduce you to the 2021 Robert E. Campbell Endowed Lecturer, Dr. Christian Hinrichs.
Good evening. Steve, thank you for the introduction. It's an honor to present the Robert E. Campbell Endowed Lecture. Bob, it's truly an honor to give this talk, and I look forward to meeting in person. The title of this talk is Winning the Fight: How Rutgers Cancer Institute of New Jersey is Personalizing Cancer Therapies to Turn Patients into Survivors. The focus of this talk is personalized cell therapy for cancer and the cell therapy program that we're building at Rutgers Cancer Institute. For our purposes here, cell therapy refers to the administration of the patient's own T cells for the treatment of cancer. For these treatments, the patient's T cells, these are the immune cells that can attack and kill abnormal cells, including cancer cells, are obtained from a tumor or blood. They're directed in the laboratory to attack the tumor, and sometimes this involves gene engineering, and then they're administered back to the patient. This type of treatment is highly personalized in that it uses the patient's own cells as the cancer treatment. The treatment is alive, and it will live in the patient and become part of the patient. Generation and administration of these treatments is highly specialized. However, thanks to the vision of the CINJ leadership, as well as a generous donation to the Cancer Immunology Metabolism Center of Excellence, CINJ is well positioned to lead this field. What initially drew me to the field is that these treatments are designed with intent to cure patients, and sometimes they do just that. The idea here is to harness the specificity and potency of T cells for therapeutic purpose. T cell specificity is remarkable. T cells can precisely kill abnormal cells without any damage to neighboring normal cells. And the T cell attack is unbelievably potent. It is driven not just by the ability of T cells to kill target cells, but also by the ability of T cells to replicate themselves to amplify the attack. So this is like a lone soldier that, when it encounters the enemy, can clone itself into an entire army. And for these sorts of treatments, we give the patients an army that then proliferates into an even bigger army. So this type of treatment is intended to kill every tumor cell the same way that T cells clear a viral infection like the flu. And of course, with this level of potency, it's important that the treatment be directed against safe targets, and this is something that we'll be discussing shortly. So the goal of our cell therapy program is to discover and develop effective cell therapies. This means that we're not only giving the latest treatments, but we're discovering the latest treatments. This type of research is important for the advancement of science and medicine, and it's also immediately important for our patients who, through this sort of a program, have early access to developing treatments that offer real hope. Our research strategy, and we're now setting up the personnel and facilities to do this at CINJ, is based on an iterative process of bench to bedside and back again research. In the laboratory, we discover new treatments. Often this involves discovering a new antigen receptor that can target a cancer in a new way. We use our process development laboratory to develop the processes for manufacturing cell products that incorporate this discovery for the administration to humans. The GMP facility manufactures these cells for the patients on a patient-by-patient basis. This allows us to conduct the first-in-human studies. When we do these studies, we build in deep translational research to try to understand why they work or don't work, so that we can then innovate a new generation of more effective therapies. And this takes us back to discovery and a new cycle of development. Now I'll discuss some of the research that we will be doing at Rutgers Cancer Institute. Much of this work is centered on human papillomavirus associated cancers. HPV-associated malignancies are incurable in the advanced stages and better treatments are needed. They include a wide range of malignancies, including cancers of the cervix, throat, and intergenital sites. They harbor the E6 and E7 oncoproteins. These are proteins that can be targeted by T cells. They also call these sorts of proteins antigens. And they're expressed by cancers and they're not expressed by healthy tissues. So this is very important because it means that for the treatment, if we are able to kill every cell that harbors E6 and E7, we will kill all of the cancer cells and cure the patient. It's also important because it means that E6 and E7 can be targeted with impunity since they're completely absent from healthy tissues. So the first cell therapy that we designed for HPV-associated cancers was called HPV-TIL. For this treatment, a metastatic tumor is surgically removed. This is the patient with multiple metastatic tumors. The resected tumor is shown here. We then grow T cells from the tumor fragments, expand these up to large numbers, and infuse them back to the patients. And what we found through subsequent research is that when this treatment works, it works by targeting the HPV antigens that I just discussed, and also by targeting other cancer antigens, including cancer germline antigens and mutated neoantigens. And this is work that we published in the journal Science. But really what was most important about this treatment was that for some patients, the tumors completely went away and they stayed away, and these patients appear to be cured. So patient 3 had widespread metastatic cancer with tumors at the locations indicated by the yellow arrows. Her cancer progressed through 3 different types of aggressive chemotherapy and radiation. This was a metastatic cervical cancer. She received a single infusion of TIL and all of her tumors went away. So here you can see before treatment, after treatment, these are all gone. She's now more than 7 years from treatment and appears to be cured. And this has been a difficult determination to make because patients with this type of disease really are not supposed to be cured. She sent me this picture after she'd been treated. It's from when her son won the Kansas State Wrestling Championship. The patient is Arrica Wallace, and we're blessed to have her here with us tonight. Patient 6 also had aggressive cervical cancer, and her cancer progressed through chemoradiation and spread to distant parts of her body. Her treatment with the HPV-TIL approach was delayed due to unexpected blood clots that occurred in her lungs, and this was related to her cancer. When she was treated, she had tumors shown by shown at the yellow arrows. And we treated her with a single infusion of TIL, and all of her tumors went away. So she's now more than 7 years out and also appears to be cured. Her case is also interesting because from studying her response, we've been able to identify genes that allowed her T cells to target the cancer. And we've created a new treatment for other patients based on these genes, and I'll discuss that shortly. So this slide summarizes our experience with HPV-TIL. 7 of 29 patients responded to treatment. 2 of 9 patients with incurable metastatic cervical cancer appear to be cured. And I say this because we gave them a single infusion of the TIL cells and all of their tumors went away and they haven't come back, and it's now more than 7 years later. This approach has received FDA breakthrough therapy designation, and it's in development by Iovance Biotherapeutics under the drug name LN-145. The research was important in demonstrating that cell therapy can cure otherwise incurable metastatic cervical cancer. Importantly, it was also the first cell therapy to cure an epithelial cancer, and epithelial cancers are the common types of cancer like lung, colon, and breast cancer. So it's important from the standpoint of showing that this type of treatment can work in epithelial cancers. However, TIL has limitations. Some patients respond to treatment, but many do not. It requires surgery, and there is inconsistency in the cell products, which are subject to patient variability in the specificity and the types of the cells that grow out of the tumors. And finally, there's a long production time. So a next generation approach is needed, and that's a major focus for our research now. So the approach that we're pursuing is to engineer the T cells from peripheral blood with T cell receptors that give the T cell the ability to specifically engage a particular cancer target or antigen. The best known of these types of gene-engineered approaches is CAR T cells, which have been highly successful in hematologic cancers. So that's shown over here on the left. But this approach has not worked in solid tumors, and one of the main reasons why is that it can only be directed against antigens on the surface of the tumor cell. So that's shown here, where a CAR antigen receptor can really only engage a cell surface protein, and these cell surface proteins represent the minority of antigens in a tumor. The vast majority of abnormal proteins in cancers are inside the cells, and this also includes the E6 and E7 oncoproteins, which are intracellular antigens. So to target HPV oncoproteins, and really most of the most attractive targets in cancer, a T cell receptor or TCR rather than a CAR is required. A TCR targets a peptide that's presented by an HLA molecule. The HLA molecules are expressed by normal cells and by cancer cells, and their job is to show pieces of proteins called peptides to the T cells. The peptide can come from anywhere in the cell, including the intracellular compartment. So, so this diagram shows the protein inside the cell. It's processed in the proteasome into peptides. These peptides are loaded onto the HLA class I molecules. These are transported to the surface of the cell. And it's this complex of the HLA molecule with the target peptide that the T cell receptor engages. And this is how T cells can target abnormal proteins hidden inside. So here's the gene-engineered T cell strategy that we're using to target HPV-associated cancer. From the patient's peripheral blood, we obtain T cells that have an endogenous T cell receptor. We then genetically engineer them using a retrovirus to express a T cell receptor that targets E7. We expand these cells to large numbers and then administer them to the patient. The engineered T cells then go to the tumors, where they target this E7 11 to 19 epitope, which is presented by an HLA-A2 molecule. Now, with this approach, we've seen some remarkable treatment responses, and I'll show you more about that now. So for patient one, a 49-year-old woman with vulvar cancer who had been treated with 7 prior systemic agents, she had extensive disease involving her lungs, retroperitoneum, right thigh, and right pelvis. You can see all of these white dots in her chest are cancer. And this disease down the middle is all cancer, and in her right thigh and in her right pelvis. You can also make out that her right leg is swollen. It's larger than her left leg, and this is because the lymphatic drain in her right leg are blocked up. And she was not able to stand for very long. So we treated her with a single infusion of TIL, and most of her disease completely disappeared. Her swelling in her right leg went down, and she was able to stand and walk again. She ended up having an 8-month response. So patient 5 was a 59-year-old man with extensive metastatic anal cancer. He had over 90 tumors throughout his body. He was previously treated with the latest immunotherapy, including a drug called pembrolizumab, a PD-1 targeting drug. And in this situation, there are no good treatment options left. Most of his tumor wouldn't fit on this slide, but you can see at the yellow arrows, multiple tumors, including bulky disease here right above his right diaphragm. All but 2 of his lung tumors completely went away. This large tumor in his kidney also shrank considerably. So for the most part, what we've seen is that when the tumors go away, they stay away, and they seem to be permanently eliminated. And this is what occurred in him. He ended up having a 9-month response. This is the last example of share. This is a patient who was a 40-year-old woman with cervical cancer. She was heavily previously treated with 7 systemic agents. She had also received a PD-L1 targeting immunotherapy. She had 2 tumors that were completely eliminated. So you can see this tumor here and this tumor here. The 3rd tumor here shrank considerably. She ended up having an 8-month response. So to summarize, the patients on this clinical trial had advanced, highly refractory cancer. 6 of 12 experienced objective tumor responses. 4 of 8 patients who had previously been treated with immunotherapy. So for these patients, there really are no options left, and prior immunotherapies have failed them. These patients experienced responses, and this is a particularly desperate situation, so it's remarkable that the treatment can work for them. Responses included striking tumor responses with complete regression of many tumors, and tumors that were eliminated generally stayed eliminated. It's beyond the scope of this talk, but we have some scientific insight into why some tumors respond and others do not. And this seems to be mostly driven by loss of the HLA complex that shows the target peptide to the engineered T cells. We're working on solutions to this in the lab, and we have some encouraging initial results. The E7 TCR is now in development by Kite Pharma as Kite 439, which gives this research a path to widespread availability to patients. And at Rutgers Cancer Institute, we will be continuing to develop this treatment with Kite and independently. And we'll be working on next generation approaches that are even more effective. We are also seeking to extend the engineered T cell approach to other types of cancer, and that's what I'll discuss next. So I previously showed you patient 6 from the TIL protocol who had a complete response to treatment and appears to be cured. And I promised to come back to her. The T cells from the TIL that we gave her targeted an HPV antigen, which is shown here in gray. But interestingly and unexpectedly, most of the cells in the TIL that we gave her targeted an antigen called KKLC1 that's shown here in the red. And this T cell receptor that is represented by this targeting is HLA-A1 restricted, which is the tissue type for about 25 to 30% of the population. And this target antigen, KKLC1, is restricted to germ cells in adults. And it's also expressed by a number of cancers for which better treatments are needed, including gastric cancer, triple-negative breast cancer, and non-small cell lung cancer. We also found that it's commonly expressed in cervical cancer, as it was in this patient. And we identified the genes that this T cell receptor, that makes the T cell express this T cell receptor. And we can now transfer these genes to any person's T cells and make any person's T cells target tumor cells and target this KKLC1 antigen in the same way that patient 6's cells did. So in this experiment, we tested this strategy in mice. So immunodeficient mice with a human cervical cancer implanted under the skin were treated with human T cells that were genetically engineered to express the KKLC1 T cell receptor that we got from patient 6. And there's a dose-dependent response where at the highest doses of the KKLC1 TCR engineered cells, all of the mice are cured. So here we're taking the genes that allowed one patient to be cured of cervical cancer, and we've shown that they can be transferred into another patient's T cells, and that these T cells can be used to effectively treat mice with a human cervical cancer. And so this and other data are the foundation for a clinical trial that we'll be conducting at Rutgers Cancer Institute. So to summarize, the KKLC1 TCR was the dominant TCR in the HPV-TIL cells that appear to have cured patient 6. Human T cells engineered to express the KKLC1 TCR can mediate tumor regression in a mouse model. KKLC1 is expressed by breast, lung, stomach, cervical, and other cancers, and Rutgers Cancer Institute will be conducting the first in-human clinical trial of KKLC1 TCR-T cells to test this approach in these cancers and others. So I'll conclude with this summary. Cell therapy can cure epithelial cancers, and we'll speak shortly with Arrica Wallace, patient 3 on the HPV-TIL trial, and the first patient to prove that this is the case. Bench-to-bedside research is driving discovery, and patient 6 is a good example of how we were able to not only cure a patient, but also derive a next generation treatment for other patients and new types of cancer from the study of her treatment response. Rutgers Cancer Institute is positioned to lead the field of cell therapy. The Center of Excellence was launched thanks to an anonymous transformational gift. It's letting us set up the cell therapy program, as well as recruit brilliant young scientists to our tumor immunology group, and to begin research that bridges immunology and metabolism. As we move forward, further philanthropic support will help us build and accelerate the impact even more. Being able to establish this program and have these capabilities has also allowed us to obtain funding through an NCI Cancer Moonshot Initiative, and also through engagement with industry, where we're working together to get these new treatments to patients. With that, I'd like to conclude and thank you for your kind attention. And we'll now have a discussion with Arrica Wallace, which will be followed by an opportunity for questions.
Dr. Hinrichs, thank you so much for all of that. Just absolutely astonishing is all I can say to that. But welcome, Arrica, to the conversation here. Why don't we start out just talking about, where were you? What was your situation when you first met Dr. Hinrichs?
So I was diagnosed with cervical cancer July 1st of 2011, and went through, standard treatment, had chemotherapy. I had 18 rounds of chemotherapy. I had 25 external radiation treatments and 6 brachytherapy treatments between the months of August of 2011 and April of 2012. I had scans that showed that treatment wasn't working, that the cancer had metastasized into lymph nodes in my chest at that point. And I did a referral at another NCI, and they told me that their trial treatment that they had was less invasive than what I'd already been given, and that I had less than a year to live. That was in February of 2012. That they told me that. They said my body would give out from the chemotherapy, that they were going to control the cancer and not cure the cancer, and that it would be the chemo that would take me. I have 2 boys. You saw a picture of them earlier, and as a mom, I just couldn't accept that. I just didn't see that was my fate, that was what was going to happen. However, in April of 2012, as my husband's picking me up off the bathroom floor and taking me to the emergency room for fluids, there was some doubt because I was pretty weak. I went to an oncologist at Research Medical Center in Kansas City, Missouri. She was amazing. And Dr. Hinrichs had actually worked with her on his surgical rotation from when he was in med school. So they had a connection, but I had no idea about that. I just know that in April of 2012, she told me we were going to take a little bit of break to give my body some time to heal. And in May of 2012 is when Dr. Hinrichs got the green light to do his immunotherapy trial for cervical cancer. So I've always believed that he was sent to me in a time that I needed it, and we've had a great connection since then.
Wow. I think we could all agree that's more than coincidence there, for sure.
Yes.
So I think you touched on this, but what did it mean to you to have access to that cell therapy clinical trial?
It was hope. I didn't know what I was going to do. I just knew that I had to keep fighting somehow, find the strength and keep fighting. And so I was pretty much game for anything. As Dr. Hinrichs said, I was the third patient, and patient 1 and 2 were very new in their treatment. I think they had just had surgeries and maybe T cells were growing in the lab or something, but very new. So, all they could tell me was that I was going to lose my hair. And I remember the doctor sitting in the room saying, that's like the only guarantee we can give you. And I had a wig on, and I pulled my wig back and said, been there, done that. It's just hair. Like, I got to live. And so I was game. I was 100% in. Whatever they wanted to do, I was going to do to fight for my life and just have hope. I mean, everything that he was telling me was going to happen with the immunotherapy and train my cells to be little soldiers and fight it all made sense. So I just thought, this is the answer. This is what's going to buy me more time to be a mom.
There is nothing, no one will fight harder than a mom who's fighting for her children. And that's amazing. So obviously we want to know, so what happened?
So, I had to go and have the surgery first, which was a little uncertain too, because it was so close to my aorta. We had to find a surgeon that was willing to do the risky surgery. And so I'm so excited to know that, the new treatments and the things that they've learned, that patients can skip that step, because that was a little stressful trying to figure that out. And I had to recover from kind of major surgery before I could even participate. But, it was hard. I was in Bethesda for 26 days in August of 2012, away from the kids, away from my family, just fighting for my life, hoping that this was going to work and this is what was going to happen. And Dr. Hinrichs was there the whole time, encouraging me. I know people think, you're the doctor, you're the researcher, you're not on the patient side a lot, but Dr. Hinrichs was there for me. And I know that made a huge difference for me and my confidence in what was happening. A funny story is that one of my main nurses tried to kick him out of my room when I was there. Before I had the second round of IL-2, I was throwing in the towel saying, I'm not going to do this for the interleukin-2, the Gatorade for the cells, I call it. But I was struggling. It was hard. And he came in to give me the pep talk that he knew I needed. And she tried to kick him out. And I was like, no, he can stay. Like, he's the one, this is his work. He can stay. But they just weren't used to seeing that. So we just had that connection. He brought his son by, knowing that I was struggling, and he brought his son by on rounds so I could see that little smiling face and little boy grin, and just pushed me through to remind me of that this is all going to be in the past one day. And we're 8.5 years in, and it's definitely in the past, but it's like it was yesterday. So anyway, he told me when I left in August, end of August, after 26 days of being in the hospital, that when I came back in September, to not be discouraged if the scans didn't show results. Like, it's still working, it takes time, just don't be discouraged. And I think he was telling himself that too. I think his pump up talks to me was also looking in the mirror saying, don't be discouraged, it's still working. But the results of that first scan just 30 days later was that my tumors were all shrinking, and some of them were even over 50% gone. So I went back to Maryland every month after that. And on December 19th of 2012, I had completely clean scans. So just 4 short months after having the treatment, I had no signs of cancer. I remember that day because I got to go around to the lab, and I got to, I got to meet all the researchers that were in the lab that don't get to see the mom that they're saving. They see my cells in a petri dish. But I got to meet them all and thank them for what they had done and their research and their hard work, because it was working and it was saving me.
My goodness. I can't even imagine the amount of tissues that would be needed for all of that.
Yeah.
Oh my gosh. That's outrageous. Absolutely outrageous. So, I mean, this is kind of a self-answering question, but I'm sure there's more to it. I mean, how has this changed your life?
It has made our entire family closer. And just the fact of, you don't take anything for granted ever, because you don't know when 3 words are going to stop you in your tracks. Anytime you hear the words "you have cancer," you just feel like you've hit a wall. Like, everything that you thought you had planned for your life is in question. So definitely, we don't take anything for granted. We enjoy all the moments. I talk to people all the time about trials and looking into trials and being an advocate for yourself to find those trials out there, knowing that there is hope when just traditional treatment doesn't work. I have one graduating in 2 days from college and one graduating in 2 weeks from high school. And I thought I wasn't even going to see him enroll in high school. So I just enjoy every moment. And now that we're going to be empty nesters, I decided to take online classes and work towards my nursing degree. I have phenomenal nurses through all of it. And I feel like that I'm here for a reason. There's a reason bigger than me and bigger than any of us, science and everything, of why I was patient number 3 that had complete cure. I say the C word, complete cure from this trial. And so right now I am a very part-time CNA at a hospice house, and just love every minute of being able to comfort and care for patients and their families during trying times. And I think I can relate to them and have a lot more empathy with them because I've been there. So I just love spending as much time with family and sharing my story with anybody that'll listen, and supporting Dr. Hinrichs and his research any way I can.
Patient becomes the survivor, becomes the caregiver. I love your heart. I love your heart that you want to give back to the world and share that. Obviously, I love this whole story. So you and Dr. Hinrichs clearly developed a personal relationship. I mean, you were going through this separate from your family, and he was the one that was there to support you during that. So tell me more about that, because I don't know that people think about the personal relationship that you need to have that level of trust at the very least. But talk more about that.
Yeah, I mean, the fact that he's on the research side of it, and I don't know if Rutgers is this way, but every time I went back, especially when I started going 6 months, I'd have a different fellow or I'd have a different person that would come and tell me my results, and they wouldn't even know my history, and that was hard. So my constant was always Dr. Hinrichs, and I don't. We just hit it off from the very beginning. Like, I mean, we'd walk in the clinic and give each other a hug, and everybody else is probably standing around going, well, I don't get a hug from my doctor. Like, what's going on? But we were a team, you know. I mean, he had the research, I had the disease. Like, we just meshed, and he needed me as much as I needed him. We celebrated the victories together. We felt the anxiety, the scanxiety before a test together. He would meet me. He'd know when I was coming in from Kansas. I'd always try to, you know, I would travel in as late as possible so I didn't have to leave home, and then have a full day of scans before clinic the same day and be heading to the airport right after that. Like, it was like a 24-hour whirlwind for me because I just did not want to be away from home, from the boys. But so he would meet me. He knew that. He knew that I wouldn't sleep. He knew that I would worry and stress about a week before the scans. So he'd meet me in radiology, and we'd talk and catch up on the kids and keep everything calm, and know that when I was going in, and he'd be looking at those scans as soon as, probably before I was even out of the machine, and texting me going, looks good, see you tomorrow. And I could just exhale and breathe. One time I went, he was gone, and I never would schedule with him being gone, but I don't know what happened. And I wasn't getting a call, and I didn't know who was going to read them. I didn't sleep all night because I'm like, oh my gosh, they're not telling me, something's wrong. I can't get a hold of anybody. But he was away, and he still was like, pulled it up, and it, like, the email came through hours later of, hey, I'm not in the office, but I looked, everything's good. I think it came through at like 3:00 in the morning because he knew I wasn't sleeping. But yeah, I mean, still when we talk, I want to know what's going on with the research, or what are we looking for as far as patients, or, he shares the exciting stuff that he just went over today, and some of it goes over my head. I'm like, okay, dumb it down to how I could understand. They don't need a surgery. Okay, great. I can understand that. But it's just, we're a whole team. We need each other, and it's just something that he gave me hope, and he saved my boy's mom, and we'll always have that connection.
Oh, goodness. I'm going to need tissue soon too. So we have one last question before we open it up to questions from everyone. But if you have one message that you could give to the Rutgers Cancer Institute community, what would that be?
I'm going to read it because I wrote it because I wanted to get through it. But research is what gives people hope. And every day that work that you do in the lab is laying the foundation to save a child's mom. I understand that you see Petri dishes, and you have goals of what that's going to look like, and you get discouraged if it doesn't turn out the way it is. Or you celebrate the victories when you do have the results that you need. And you feel like that maybe it's failed because it didn't work in a patient, but it's not failing. It's learning and adjusting, and knowing in that whole time that patient still has hope, and you're giving them time. Just having the fact that you are researching, I could have fought through more chemo for another few months while you guys were in a lab researching something that would save me at one point when I needed something different. So just the luxury of time that you take for us on the outside while you're in the lab is huge. You will get the big wins. I don't know if Dr. Hinrichs believed that until we came through. I know he hoped for it and prayed for it, but you will get the big wins and be able to celebrate the 4-letter C word of cure. But, we appreciate all the hours and all the hard work and all the frustrating times of the research that's going on more than you can ever know. And I don't know if supporters are on here, financial supporters or not. But without the financial investment, none of this would be possible. You're supporting hope. Your money is going to hope and giving the luxury of more time. And it takes all of us. It takes from the patients, the nurses, the doctors, the researchers, and the supporters to ensure that we're all going to win the fight.
Thank you so much. Thank you for sharing all of that, being so open about everything.
Sure.
Dr. Hinrichs, that's amazing. I'm sure you have some, responses to what Arrica shared with us. I mean, what in the work that you do, not everyone can be cured, but that is what you're doing every day. What keeps you going through all of this?
I never thought about it. For me personally, I think that I'm so internally motivated to go up against this problem that I never would stop that much to think about why. It's an awful problem. It touches all of us personally. I mean, there's just like no way to make it too far through life without encountering cancer close to home. And there's just a compelling need to do better with the treatments. And I think there's also a compelling need to take kind of big swings at it. And by that, I mean, really innovative new approaches that are designed with intent to cure the patients, that have less of an incremental design and more of a, making a big difference, curing someone who's incurable, sort of a strategy. And so I find discovering and developing those sorts of treatments entirely compelling. And I don't know that I could tell you why, other than just that that's what they are.
Oh, I love it. I love it. Arrica, did you want to comment on that?
I think he's driven that way, but he definitely takes pride in his work, and it's amazing. I mean, his why is to be able to celebrate when we have the wins. I think that's why we're so close, is that without all the hours and all the work and kind of a change in his path professionally and stuff for him too, it was all for a reason. It was all, again, I go back to higher than science, that his path went away that ultimately led to curing me. And I know scientifically we can't say cured, but I mean, 8.5 years of clean scans, like, we're going with cured. So.
Sounds good. I'm here for it. So we have some technical questions from the audience. This question, it says, what are the potential barriers to develop off-the-shelf allogeneic T-cell TCRs? That is a much smarter question than me.
So this is kind of a sciencey question, and I have to give kind of a sciencey answer. So, the biggest obstacle with allogeneic T cells is that means where instead of making each cell product for each patient from that patient and then for that patient, can you make a bunch of cell products for different patients from one individual, maybe a normal donor, and then be able to give those to each of the patients? And it's a fantastic idea because it would greatly simplify delivery of the treatment, because you don't have to make a cell product for each patient as they come in the door. The greatest scientific obstacle is that the allogeneic cells, the cells from another person, if they go into you, your body, your immune system will recognize that those are foreign, and it'll kill them off. So the scientific obstacle is in figuring out how to make those cells essentially invisible to the recipient's immune system so that that immune system doesn't identify and kill them. And the leading strategies for doing that are related to gene engineering to take out expression of the key genes in the cells that are given so that you can make them invisible. And scientifically, really, we're at the point where we can do that, and these can start to move forward. I think that from my point of view, as someone who's trying to discover and develop completely new treatments and take them to the patient, and is trying to kind of break new ground from a scientific principle sort of a standpoint. The first way to tackle these problems so far has been using the patient's own cells, because there you can test principles like whether the whole treatment approach can work at all. And then the next steps are to make it more easily deliverable by doing things like using allogeneic cells.
Thank you so much. The next two questions are somewhat related here. So they're asking, do you see this treatment becoming a first-line therapy even in early stage disease? And then the second question is asking if patients would have to exhaust all traditional therapies before qualifying for immunotherapy. So
A common theme in both questions, sure. And they're great questions, and it's exactly along the lines of what we're thinking as a clinical development team. And probably, part of, a limitation right now is that we're treating patients who have already been treated with everything, including immunotherapies. And in their tumors kind of escaping or surviving in the face of these immunotherapies, the tumors are getting conditioned to be very resistant to immunotherapy. And it makes a lot of sense to move this sort of a treatment up earlier. And we're definitely doing that. So we have plans to open at Rutgers a clinical trial with the E7 TCR approach, where we'll be trying to treat patients actually before they get any therapy. And you hate to talk about expectations from science, because really it's kind of an exploration, and often expectations aren't what happens. But really, just from a logical standpoint, it seems like they ought to be, very highly effective if we can get the patients before their tumors have been treated with all these other treatments, and before they have, literally 90+ cancers.
So talking about all of this, question for you, Dr. Hinrichs, what drew you to come to the Cancer Institute of New Jersey?
Oh, that's a great question. So, CI&J is just perfectly positioned to do this type of treatment. So, the question that I'm constantly asking myself is, how can I make the most progress the fastest? And where can I do that? And I think the answer became CI&J. And this is driven by vision of the leadership for a cell therapy program at the Cancer Institute. And it's that same vision that's made sure that we have a GMP manufacturing facility on site where we can make the cells for each patient and give them to each patient, to have a process development lab on site so that we can develop the processes for making those treatments. And of course, very importantly, we have this patient population that is so much in need. And, just how Arrica was articulating how she needed me and I needed her, it's really the same situation for this entire patient population. And so that's why I'm at CI&J. I think it just has all the right elements to do this.
Terrific. I have someone here specifically asking how they can become part of your research team.
Oh, email me and we'll talk about it.
Yeah, absolutely. I don't have any more questions from the audience. By all means, if you have any more questions, please throw them in the Q&A. But we are coming up to the top of the hour here. And so I think I just wanted to ask if you have anything else, either one of you, that you would want to share here. I think this has been an amazing, extremely educational, but also, like Arrica said, so much hope when in general people think of cancer. The hope is not the first thing that comes to their mind. It's just not at all. And sadly, some people never find that hope, but this is exactly what you are in the business of creating, and it's amazing and wonderful. So anyway, so I'll stop talking if there's anything you want to leave the audience with before we go.
Well, thank you for the opportunity, and really, Arrica, also thanks for coming to speak. As I was trying to think of what to speak about for this talk, I was thinking a lot about kind of the theme of personalized therapy, and how, of course, the treatments are personalized, but the relationships that we develop and the relationships that go into their development also are obviously very personal. And you know, having Arrica here, I think, is a chance to kind of illustrate that. And of course, I'd just love to see her, and you know, we continue our work together.
Well, I think that is all we have for this evening. Thank you all so much for joining us. Thank you to Bob Campbell for all of his service and support of this endowed lecture. Thank you, Dr. Lee, for his remarks and leadership. Thank you, Dr. Hinrichs, for sharing your amazing work, and Arrica for sharing your personal story and just your heart. And thank you to each and every one of you for tuning in. We can't thank you enough. Have a wonderful evening.